Hello everyone, I am Norilyn M. Cardona. Mayet Montederamas and I are going to report the human gene therapy. Because some mutants can alter gene function and thereby cause disease, geneticists are actively pursuing the goal of using normal, cloned genes to compensate for defects in human mutant genes. Gene therapy is the introduction of cloned genes into somatic cells or the modification of existing genes in order to treat a disease. It is a potential method for treating a wide variety of illnesses. Many current research efforts in gene therapy are aimed to alleviating inherited human diseases. Over 7,000 human genetic diseases are known to involve a single gene abnormality. Familiar examples include cystic fibrosis, sickle cell disease, and hemophilia. In addition, gene therapies have also been aimed to treating diseases such as cancer and cardiovascular diseases, which may occur later in life. Some scientists are even pursuing research that will use gene therapy to combat infectious diseases such as AIDS. Even though a large amount of research has already been conducted, success has been limited, and relatively few patients have been treated with gene therapy. Gene therapy may involve the introduction of cloned genes into human cells in some forms of gene therapy. A key step is the introduction of a cloned gene into a person's somatic cells. This is a difficult challenge because it requires that many cells of the person's body take up the cloned gene and express it. Otherwise, the disease symptoms will not be corrected. The techniques to transfer a cloned gene into human cells can be categorized as non-viral and viral gene transfer methods. The most common non-viral technique involves the use of liposomes, which are lipid vesicles, which is seen in figure 22.15a and to be discussed by Mayet Montederamos. The DNA containing the gene of interest is a complex with liposomes that carry a positive charge, for example, cationic liposomes. So, the DNA liposome complexes are taken into cells via endocytosis, in which a portion of plasma membrane invaginates and creates an intracellular vesicle known as an endosome. So, the liposome is degraded within the endosome. Therefore, the DNA is then released into the cytosol, imported into the nucleus, and then integrated into a chromosome of the target cell by recombination. An advantage of gene transfer via liposomes is that the liposomes do not elicit an immune response. A disadvantage is that the efficiency of gene transfer may be very low. A second way to transfer genes into human cells is via viruses. Commonly used viruses for gene therapy include retroviruses, adenoviruses, and parvoviruses. The genetic modification of these viral genomes has led to the development of gene therapy vectors with a capacity to infect cells or tissues, much like the ability of wild-type viruses to infect cells. However, in contrast to wild-type viruses, the viral vectors used in gene therapy have been genetically engineered so they can no longer replicate within the targeted cells. Moreover, the genetically engineered viruses are naturally taken up by cells via endocytosis. So, the viral coat disassembles and the viral genome is released into the cytosol. In the case of retroviruses, the genome is ribonucleic acid, which is reverse transcribed into deoxyribonucleic acid. The viral DNA, which carries a gene of interest, is imported into the nucleus and is integrated into a chromosome of the target cell by recombination. Most importantly, a key advantage of viral vectors is their ability to efficiently transfer cloned genes to a variety of human cell types. However, a major disadvantage of viral mediated gene therapy is the potential to evoke an undesirable immune response when the virus is injected into a patient. Also, the inflammatory responses induced by adenovirus particles, for example, can be very strong and even fatal. Therefore, much effort has been aimed at preventing the inflammatory responses mediated by virus particles. So this includes the use of immunosuppressive drugs and the generation of less immunogenic viral vectors by further genetic modification within the viral genome.